Hello, and welcome to my presentation, a survey of the macroinvertebrate community living in the Quequechan River, Fall River, Massachusetts, fall 2022. My name is Melinda Amarantes. I am an honors student and biology major at Bristol Community College. My faculty sponsor is Professor Mary Rapine, Professor of Biology. We have been working on the Quequechan River Project with Professor Robert Rack, who is the Environmental Science and Technology Coordinator at Bristol. The goal of the Quequechan River Project is to assess the water quality of the Quequechan River. Professor Rack's team assesses the biochemical parameters of the water while we categorize, enumerate, and document the benthic macroinvertebrates. The Quequechan River is a 2-mile-long river that flows northwesterly from the Watuppa Pond through the city of Fall River and into the end of the Taunton River at Mount Hope Bay at Heritage State Park, which is also known as Battleship Cove. The word Quequechan means falling river in the native Wampanoag language, which is where the city of Fall River gets its name because it was built on top of the Quequechan River. Currently, the water of the Quequechan River is not potable due to many sources of pollution such as unchecked sewer runoff, runoff from factories that once operated along the embankment of the river, and of course, illegal trash dumping by the city's residents. Water of a river is in constant movement, which means that the water in your first sample will differ from the water in your second sample, even if the samples are only taken a minute apart. Benthic macroinvertebrates, however, are fairly stationary, which means they are excellent indicators of pollution, as some organisms are more pollutant tolerant than others. So by studying the macroinvertebrates along with the biochemical parameters of the water, it gives us a better idea of the true quality of the water in the river. I have been taking samples from Site 3, which is a location on the Quequechan River Rail Trail located behind the Fall River Police Department. When taking samples, I use a sampling tool called a dredge to collect sediment from the benthic layer of the river. The sediment is then placed into a 5-gallon bucket that has been marked with liter increments. Inorganic material and large plant debris are removed from the sediment, and the sediment is mixed to allow for a more even disbursement of organisms. I then take a sample from the mixed sediment, filling a 1-liter plastic bottle 3/4 of the way full. The sample is then brought back to the lab where it is processed. In the lab, we pour a small amount of sample into a plastic tray and dilute it with deionized or distilled water. This way, it makes it so that it is easier for us to see the organisms as the sediment is very dark. Once we have spotted an organism, we remove them with pipettes or forceps and place them into a petri dish to view under a dissection microscope. If further magnification is required for identification, then we take the specimen and place it on a depression slide and view it under a compound microscope. In fall of 2022, I personally identified and cataloged 12 different types of macrobenthic invertebrates, including non-biting midge larvae and biting midge larvae, dragonfly nymphs, amphipods, copepods, cranefly larvae, flatworms, ostracods, bladder snails, pygmy back swimmers, water mites, and nematodes. The Quequechan River Project is still a very young project, and we have yet to collect enough data to be able to fully understand the quality of the water in the river. I plan on continuing with this research until I graduate in the fall of 2023. Until then, my goal is to continue working on collecting samples, identifying, enumerating, and documenting the macroinvertebrates, while also creating a field guide for future Bristol Community College students to use as a guideline while continuing this very important research. I hope you've enjoyed my brief presentation on the work I have been doing in the Quequechan River. Thank you so much for your time.